trying to look into that how the integration of the genomics and proteomics would help us in better understanding. And I think by till now you would have appreciated that how the genomic level changes are translated at proteome level changes, and how you can detect those, and you can understand the molecular basis of those changes in a better way. But for going to the steps or the integration of the steps and the data size by looking at the data, so it goes through a lot of steps and goes through a lot of uh, issues in the main in the phase of the challenges, like the generation of the data, sample preparation, then understanding the data. So all these things are would be or quite necessary to understand that how you can you need to go through it. So now, for example, if we look at a proteomics approach, so if you just look at a case kind of a case study or kind of a thing, that how you will uh, understand. So, for example, if you take a proteomics study, proteomics for any particular disease. So now in the genomics level, you can identify your variants and identify your SNPs. Now in the proteomics study, so we have till now gone through the steps for your starting from your sample preparation, proteolytic enzymes, then your mass analyzers in mass spectrometry based proteomics, then your that how you will try to understand and analyze your data. So the steps like normalization, then statistical test, then machine learning based approaches, your unsupervised learning or the supervised learning. So all these we have covered and we have seen till now, which you all can refer as well. So now if you think of a full-fledged large scale proteomic study for any particular disease. So for example, here you will take your biofluids. So you can take biofluids like serum, plasma. So the main power of the proteomics lies that you can take any kind of your biospecimen, and then you can try to find out if there are any relevant changes because of your diseases, and if you can find out some protein markers out of it, or that can be treated as for your different diagnostic or the prognostic purposes, so that you can take and try to find out. So you can take any kind of biofluid, any kind of the tumor tissue. So then you will do a global proteomics. So from here you will do the sample preparation, you will extract the proteins out of it, lyse to it, and then so we have discussed thoroughly. So I think you all can refer if you all just cannot recall, or otherwise try to recall that we have discussed thoroughly that what we are doing here in the proteomics is mostly the bottom up proteomics, where we take the protein, digest into peptides, try to identify the peptides, and then try trace back and try to identify that what proteins we have identified and what is the expression level of that particular protein in our disease state, normal state, or the treated state, healthy untreated state, and different conditions. We try to compare it. So now after you have collected your any kind of biospecimen and bio fluid, so on the basis of your sample preparation, that how you would prepare the samples, you will do the protein extraction followed by proteolytic digestion. So proteolytic digestion will give you the peptides, then you will try to do the LC-MSMS, and then you will do the peptide mass spectra. So now in this, what is happening is that you have got, then you will find your raw data, and you will do your primary analysis, then the statistical analysis. So now what is we seeing that now, for example, you have 100 samples of for any particular disease or patient samples, so clinical samples. So you will do a large scale MSMS proteomics or a discovery based global proteomics. Now I think you should understand that now once you've got the protein expression levels in your control and your diseased condition. So now what you will to do is that the panel of the proteins, either through the statistical methods or the feature extraction or different machine learning based tools for your feature selection. So you will now identify a panel of proteins or the number of proteins which are showing differential regulation in your disease versus control state. So now from this step, and this step, we are are we all have discussed in detail, which I think if you all don't recall, can look at the previous recordings and can understand. So each and every step has been discussed thoroughly, like why MSMS, how the peptide mass spectra is generated, what are the different mass analysis and the functionalities of the mass spectrometry, and then the different statistical ways, different t-tests to identify your proteins that which are differentially significantly differentially regulated, or you can use your different machine learning based algorithms features to select out the features for diagnostic purpose, early diagnostic purpose, for prognostic purpose, that your patients will become severe or will become aggressive or not. On the all things you can do that. So now we are at a stage. Now we have done a global scale proteomics. We have got a number of proteins we have identified that are really getting distinguished in the two different states. So now what is the next step? Because if you just discover a set of proteins or a panel of proteins, you cannot go forward and can propose it to be in a clinicals or in clinics directly. So you need to prove or a kind of a validation or a next layer of validation that you need to perform, so that you can say that okay, so the proteins that we have identified through the global. So here one concept what I had discussed, that whatever in the statistics, whatever the main concept behind rejecting our this null hypothesis, that the whatever data we are finding or whatever difference we are observing is not by chance and is actually the biological difference, or is not any technical artifact. So there are some case studies, there are some case studies, and there are have been some stories, means there have been some incidents which has happened, is that there have been a protein which has shown you a very good differential regulation between the diseased and the control state, but when it was applied, or it was that protein was tried on a different sample set of cohort, it was not coming that much significant. The reason is that during the collection in the tube for one set of sample, for one batch of patients, it became as it is, that in that, for that, due to that reaction in that tube, that protein was getting modified or something was happening, which led to your identification of the differential expression in those particular protein in that patients. So that's why it is becomes very much crucial to have a use a different method, add a different set of things to become truly sure that what we are doing is not artifact. So we also had discussed that we whenever we are trying to identify any kind of protein or biomarkers or anything, when we claim or when we try to propose that it is your proposed biomarker or a proposed marker that can segregate your proteins of interest and all, then we should be very much careful about the false positives, because if you take that particular proteins which is actually not segregating the grades or segregating the control and the disease state, and you are taking this to clinics, it would be very much detrimental if it is so the controlling the false positive is very much crucial. So here that's why the steps comes of our another thing, that we need to validate it. So the validation should be on a larger number of samples than your discovery set of samples. It should be ideally it should be on a different cohort of samples. A set of cohort of samples that we have used for discovery validation should be used in a different cohort of samples for your target validation. Another thing is that it should be the ideal thing would again be a blinded kind of validation. So if you are predicting certain kind of proteins or markers that are segregating your control versus tumor, so now you will be blindly followed trying to validate those markers, that whether that you won't know that it is control or tumor, or let your marker or your data try to identify and then see that how much it is correctly predicted. So these are actually the steps through which, after which you can go and say that you have discovered a biomarker, or for the these are the different steps that are generally followed for biomarker discovery. Now another thing is that the different analysis which you can do is the metadata, or the analysis or the data analysis from your literature. So why it is quite important? So for example, so I am hoping that you all have heard about the term that reproducibility. So in science, what matters the most is the reproducibility. So what is that? If whatever any experiment that you have done should be if that all conditions are met, so then it should be reproducible by anyone. But another thing that we need to understand, that whatever, so I think in the false discovery rate we have said that it is a kind of in the field of proteomics, or in any kind of, for example, in the RNA-seq data as well for the gene expression study. So whatever we are doing is the kind of a multiple hypothesis testing. So what does it mean is that the set of proteins that you are saying are claiming to be a biomarker, or claiming to be a set of significantly dysregulated markers. So these kind of markers, what it generally you can see that these kind of markers are again tested in the different labs using different cohorts of samples. So the set of proteins that you are saying that it is differentially regulated on a set of population can or cannot be the same in a different cohort of population. So it is a global, is it because it this can be of due to various reasons. For example, first major factor can be environmental reason. Environmental factors can induce certain changes in a certain set of populations, whereas in persons or the patients in a different set of environmental conditions have different sets of markers or different set of proteins. So these literature search, so you know that the huge amount of data is getting generated and reposited every single day, at lots of data in all the fields of omics like genomics, transcriptomics, proteomics, metabolomics. So you name anyone, data is getting generated for each and every disease and is getting reposited. So now it becomes crucial also to try to find out that what the markers that you are saying or you have identified from your experiment, how it is coming or how it has been reported in the literature, to try to find out that whether your data is getting matched, what is your global level, so which markers are coming common across the globe, across the different labs have reported. So what are those common markers that you are trying to find out and retrieving? So for this, what you need is that you need to identify the different go through the different literature, try to find out the data. So where you will find out, so there are different repositories are there. So how then you need to curate your data, and then finally you try to match it with and try to find out or the report the global proteins. So whatever till we did now was the global proteomics workflows with your, you generated your data, you analyzed your data, and you have now your feature selection. You have your different features or the different proteins. So now what you would like to do is that validation of those proteins and the data storage and the accessibility. So in this today's class, we will mostly try to focus on the validation, that how the proteins can be validated, mostly focusing on the MS based methods, and try to find out that how the data storages are there, what are the popular data storages and can be accessed and can be analyzed. So you don't need to be present in lab or anywhere or use different uh, heavy software or something, but people have tried to help you out, the researchers, the curious persons, to with the different automated software and the computation techniques, so that you can, if you are curious enough or you are curious enough to know for a certain disease or a certain thing, so you can try to find out or access those data sets, use public data analysis softwares and servers, and try to find out that what is the markers and what is the expression level, So I think one I have already given, like the CBio portal, which has already analyzed and has kept all the mutational status and all. So where you can try to find out for any particular disease the status of those genes and how it is getting correlated, and the known popular gene markers for certain cancers, how they are behaving in different fields. So now today we will mostly be focusing on the validation of those proteins, how to validate those, and the data storage and accessibility. So now we have the list of proteins. So if we tell you the validation, the term validation, so the validation can be divided into two things. One is your antibody-based validation, for the conventional or traditional method that we generally being used, uh, your Western blot or your MS-based validations. And in the antibody-based validation, you all know, like you follow Western blot. ELISA is one of the most Western blot and ELISA are very much popular techniques that are used for validating different proteins in your sample. So if you are saying that, for example, a particular protein X is getting upregulated in your severe malaria patients, for example. So now how you would say that in a quantifiable manner? So ELISA will give you that opportunity to quantify at your picogram level the protein, and you can with the standard protein concentrations you can make the graph and then finally try to correlate with the amount of the protein concentration in your sample. So that will be a validation. So this can be an experimental strategy where you have done a MS based analysis. For example, you have taken the types of patients having malaria who have got severe and who have got non-severe. You have run your MS-based analysis. You have identified the proteins markers, and then what you did is that you validated it in uh, ELISA, and you have shown that the actual quantifiable level of the protein. So global in using MS-based method, and this using antibody-based method, where you can see the show that this particular protein is showing the same trend wherever you have used your whether you are using mass spec-based method or you are using ELISA enzyme-based method. It is showing the same trend. So this can be one. Other things are like immunofluorescence or immunohistochemistry. All these techniques can be used for your using antibody-based method. You can find out. Now another thing is that so now we will be looking into it, that what are the pros and cons of these methods, and how what are the that antibody-based method, what are the problems and what are the advantages. And another is our MS-based. So in the mass spectrometer, so I hope you all are recalling the different mass analyzers. So if not, can revisit and try to look at those mass analyzers as well. So now we have different mass analyzers. So in the global set, whenever we are doing a discovery set, we use a different kinds of parameters and a different kind of technique to try to find out all the proteins in the sample. So as much ions can be generated, as many ions are generated, and as many ions are detected. So one's goal or one's thing is to try to find out those particular kind of proteins. But in another method is to find out that in a target that only those particular proteins or those particular peptides ion can only pass out through a filter and can be detected in the detector. So this is another method through the mass spectrometry based method only. So if so how this would work is that if you're in the actual set the samples, so you will specifically select those particular peptides and select those particular ions and the ion filter, and will ask the instrument to detect only those, and the others uh, would not be monitored. So this is another method for the validation. So now we will look into it in detail. So antibody based method is the major thing is that it is low throughput. So first thing is that whether you do ELISA, you do Western blot or immunofluorescence. So the thing first you need is a antibody. So you have to identify your particular proteins of interest. If antibody is not available, then you have to either raise the antibody, then try to then perform the experiments. So it is also very much that it is labor intensive, and you can only monitor in your samples only one proteins at a time when you do ELISA or Western blot. So it is not very much high throughput way, and the cost associated with your antibodies generation, then you looking at those values and performing the experiment. So these are the some of the limitations of your antibody based method, but as you all know that it is quite robust and reproducible, your ELISA and Western blot, and that's why it is being used, and it is also used in the different diagnostic purposes as well. So in ELISA is used for different diagnostic purposes for detection of different diseases, but these are some of the limitations associated with antibody based methods. So now if you have, for example, if you have got 100 proteins or 100 proteins panel for segregating between two severe patients, then you cannot do test it out 100 ELISA one by one, or you cannot it is not means it is not very much convenient to do that. And as well as if you for leave 100, if you have 10 12 panel of proteins as well, so it is not very much convenient to routinely use or monitor a large number of proteins. So for example, here I just given you one example. So I am hopeful that you can always visit this. So human protein atlas is that which provided you the tissue-based map of the human proteome. So they used an antibody-based protein profiling using conventional and multiplex immunochemistry. So that how your what is the map of your proteins in the particular tissue. So this is one of the major thing where antibody-based methods have been used to determine the proteins localization and the expression level at the protein level. So this is one of the use which has been done extensively using the your antibody-based method. So this we need to now I try to find out means or ways so that we can do it in a much high throughput manner with a high in a quite a reproducible and robust manner, as well as we need to validate our set of proteins as well. So now comes the MS based method, which is quite of a high throughput because you can monitor a set of peptides and set of proteins in a kind of an automated manner. So you need to prepare the samples as you are preparing the samples for your global MS based proteomics. So in the targeted proteomics also you can try to find out your do the sample preparation, and then uh, will your instrument would acquire your data and quite kind of robust and reproducible. So this is an example where MRM or multi-reaction monitoring based, which we will discuss that how in detail how it is performed and how it is followed, has been used for the validation of the protein biomarkers in brain tumors. So this is kind of a paper which has shown that how mRNA-based targeted assays can be used for the validation of protein biomarkers in brain tumors. So you can see that the same samples are taken, it is digested, the workflow remains same, but here the mass analyzers that are used is different. So so if you remember, or I would suggest you all to go back and check the mass analyzers thing. So then you will do the data analysis and then try to find out that what is the level and what is the level of those finally your. So here in this method, another thing is that you can do both relative quantification as well as your absolute quantification. So if you have your synthetic, so you know that you are monitoring, for example, in the mass spec. So since we are doing your bottom up proteomics, so we are monitoring the peptide. So if you are very much sure about a particular peptide or the proteins which is coming to be differentially regulated. So you can generate your synthetic peptides and try to find out in the unknown standard what is the quantification, and then in a standard curve, make a standard curve, and then try to find out that in the unknown concentration what is your absolute concentration in your samples. So here you can use both absolute and the relative quantification. So this is was the method. So in the targeted proteomics, it has been in the year 2012. So it has came as the method of the year, and it was published in the Nature Methods. So it has been told that it is for that year. So that it was one of the highly is invented technique which you can lead you to validate your things, validate the SRM or the by the, as I told that you can see, right, multiple reaction monitoring. So SRM or MRM based technique for your validation of those proteins without the use of antibodies. The conventional use of antibodies that is being generally done, that was generally done till now, that it is also analysis of, for example, you can see that analysis of a pre-selected group of proteins delivers your more precise quantitative sensitive data. So in this way also one can try to find out and validate those proteins, except the antibody based methods as well. So now these are the your three kinds of uh, these are the two kinds of uh, your techniques which through which you try to validate. So let us go one by one. So this is your selected reaction monitoring or multiple reaction monitoring. So the paper that I just showed you was used this method, single reaction monitoring. So there, what how the mass analyzers are used? The mass analyzers are used are two quadruple. So this kind of your setting is known as triple quadruple, where you have first quadruple and another third quadruple and the collision cell in between. So what it is done? So if you remember that the uh, principle of quadruple mass analyzer, that it has four rods with two in opposite charges, and it acts as a kind of a filter which allows the particular ions of having particular M bases to pass through. So now here in this first filter, you would pre-select your filters, your ions that can pass through it. Then it will be collision in the Q2, that in the it will be fragmented here in the collision cell, and in the third quadruple, the pre-selected this ion and the daughter ions which are generated here would be allowed to detect in the mass spectrometer. So you can see that whichever fragments that is being allowed to have been goes to the detector and gets detected. So in this configuration of this is a triple. This is known as your triple quadruple uh, mass spectrometer, where there you have two quadruples with one collision cell, so which allows you to go and detect in the mass spectrometer, and allows you for identification of only those particular peptides or proteins that you are allowing it to. So the means that you are of that is of your interest. So this is your selected reaction monitoring or multiple reaction monitoring. So it is known as multiple reaction monitoring when you are looking at multiple transitions or multiple uh, uh, transitions at a time. So that it becomes your multiple reaction monitoring. So now another thing is that the another. So that's why the understanding or the concepts related to mass analyzers is somewhat needed for you to better understanding that how you can design your experiments for different purposes. So now another thing is the parallel reaction monitoring. So these are all your mass spectrometry based methods for your validation of those particular proteins. So now here you will have the first filter as quadruple, and then you will in the collision cell it will be fragmented. All your the whatever ions you have the precursor you have let it go, it will be uh, fragmented in your collision cell. But here, except of another quadruple, you will have it will be replaced by orbit trap. So I think you all remember that there is another mass analyzer, that is your orbit trap. So I think you remember that we had discussed about time of flight, ion trap, quadruple, and orbit trap. So here it is orbit trap. So what it is doing is that it will then look at all the ions of that particular precursors that has been fragmented, will be monitored. So here again, this in this what the difference between your SRM and PRM is that majorly here you will define your precursor as well as product ion, and when and it will detect only those particular precursor and product ions. Only particular you will define that also, which is known as your transition, which we will look into detail. But in PRM, what it happens is that for a particular precursor, all the fragments will be monitored in the orbit trap and will be detected. So this is your major difference between your SRM and the PRM. Whereas the DIA or data independent acquisition is a kind of another technique, which here mostly it is not related to much related to your uh, targeted experiments, where DIA you can try to find out your different in the global scale as well, where you will try to actually in the global proteomics, these are I don't know if I don't remember if I have discussed or not. So there are two methods. One is your data dependent acquisition method, and another is your data independent acquisition method, where on the basis of the ions of the precursors, if you uh, if you allow, if in the orbit trap, if you detect only the top n abundant precursors, then it is a DDA method, and if you allow all the precursors to get monitored, that is your data independent acquisition. So that is not much related to the targeted, but the major two ways that we generally targeted proteomics that you will validate is your SRM and the PRM. So major thing is that you in the SRM you will give both the not only the precursor but as well as the product ion. You will include that for it to get monitored using two quadruples and in a triple quadruple setup, which is the mass analyzers is known as your triple quadruple instrument, but actually which contains your two quadruples. And in the PRM, you will have particular precursor and you will fragment it and you will detect all those precursors. So that's why you can see that here there are selected spectra, but here you will detect all the spectra in the PRM based method. So this is the two popular with very much highly method for your validation other than your antibody based methods. So here we will look into in detail. Firstly, the selected reaction monitoring or multiple reaction monitoring. So here, as I mentioned, that you have your triple quadruple mass spectrometer. So just remember, so this in the triple quadruple, so one Q1 and the Q3, and is separated by your collision cell. So here you will select your precursor ion, then it will go into the collision cell where it will be fragmented, and then you will again select from this. So you are selecting precursor, you are selecting the product ion, and finally it is going to the detector. So here you are selecting both your precursor, both your product ion, and it is getting into the detector, and you are then detected. So fragment 1, fragment 2. So this will give your particular transitions. Now in the PRM, you have an orbit trap mass spectrometer. So first difference between your SRM, MRM and the PRM is that what kind of mass spectrometer will you use. So whenever if you are to design or if you are to ask to design an experiment, or that you will be using SRM, MRM or PRM. So first what you have to understand that what type of mass and instrument would you use for doing. So for MRM, SRM, you will be using your triple quadruple mass spectrometer, but for a PRM, you will be using orbit trap mass spectrometer, where you will be looking for all the fragments of the precursors and then try to identify. So here you will be precursor ion Q2 fractionation and all those fractions. So here you are selecting that particular ion. So for example, these small circle violet ions you are selecting. So you are selecting precursor and the product ion. Here you have only the precursor products, so all the products. So big violet, small violet, everything will be going into the orbit trap and detect. So the number of fragments that you get in SRM will be less, whereas in PRM it will be much more. So now if we look for the selected reaction monitoring or multiple, so I use the term is your transition, right? So now what is your transition? So specific pairs of mass to charge values. So just try to understand here carefully. Specific pairs of mass to charge values associated to the peptide precursor and fragment ions. So as I mentioned that both precursor and the mass ions, so specific pairs of that. So the Q1, Q3 pair. So that pairs of mass to charge values associated to your peptide precursor and your fragment ions are referred to as transitions. So what is your transition? That is the Q1, Q3 pair, the specific pairs of mass to charge values that is your associated to the peptide precursor and fragment ions are referred to as your transitions. So you have the precursor, targeted precursor M by the isolation using first quadruple. Then you will have the collision cell where you will fragment and then selected fragments. So this is will get the MRM MS spectra. So this is known as these specific pairs of M by Z ratios or values are called your known as your transitions. Yeah. So any question till now, or any doubt or anything? No, right? Okay, then we will move forward. Yeah. So this is what transitions for a triple quadruple experiment for a Q1 and Q3 to understand that what is the precursor and then what followed by your fragment and then selection of those particular fragments. So now for SRM, MRM assay development, so what we need certain, uh, we need to identify and understand that how we can perform if we are asked to develop or the SRM MRM assay. So when we will do, so there are 2-3 questions that when we will do and how we will do. So firstly, I have already told that you need to understand that what kind of instrument you will be using. You cannot use any instrument for using any kind of particular technique that you need to understand. So firstly, we need to have your discovery proteomics data, because how will you derive your proteins or peptides of interest? So you cannot try to identify all your proteins present in a sample. So if in a human, if you have 20,000 proteins and try to identify 20,000 proteins using this, so this is not going to happen. And why that I can say that though it happens in a high throughput manner, but it has also certain limitations of monitoring the peptide. So you know that you cannot bombard your uh, uh, you cannot bombard your detector with all the ions at a time, because then it will be overlap. Secondly, if or many proteins will have a specific set of time when they will get eluted out, and then there will be a continuous overlap, and it will be difficult for you to identify those particular proteins. So you have your discovery proteomics data. Either you have done your discovery proteomics data, and then after the analysis you have selected out the particular proteins or the particular peptides, and then can identify those particular things. That can be one. Second is your proteomics data repositories. As I mentioned, that from the data repositories you have analyzed, you have downloaded and analyzed your data, and then have found out certain markers. So now the interesting part here is that the proteomics data repositories will be containing of the data of the different uh, studies from different labs. But for example, they have done your analysis between two grades of a patient, but you are now interested. So what you are interested now that okay, so now I want to check that you have found out that there is a distinct in the difference in the gender or in the age of the patients, age differences in the patients, or gender differences or gender biasness for a particular disease, and you want to find out the what is the difference between a male versus female, or between a younger age population or a older age population. So you can always go through that, download that, try to find out the analyze the data, find out those differences, and then you are trying to validate in your samples if you have certain clinical samples, and try to came up with a certain hypothesis and then trying to validate it out. So that is feasible, and that can be required, and that you can do. Or else what is that using the literature there have been reports of the any particular gene or a particular protein, and then you can try to find out. So this is one kind of thing in the SRM. So firstly we need to understand the proteins of peptides. Now if we look at the technicalities, that what things should be needed to try to set up an SRM or MRM experiment. So first thing is that we need to have a signature or a proteotypic peptide. So so as you know, all of you know that we are not looking at a set of only whole protein. We are not looking at a particular protein or a whole protein. We are looking at a peptide. We are not looking at protein, rather we are looking at peptides. So now the peptide should be unique to the protein of interest. So this is a good concept. So I will show you that how you can ensure that the uniqueness is there, as well as there is one tool where one server called Nextprot. So I think you all have heard of Uniprot, or aware of Uniprot. So another tool is their Nextprot, where you can try to find out that the peptides are unique or not. So what do you mean by unique peptides? So for example, you have a protein, right? Now protein has the amino acid sequences. So you are using, for example, if we are using trypsin and you are cleaving that particular peptides, now all those particular peptides might not be unique to that particular protein, means the sequence of that particular peptide may belong to different proteins, that it will be called your shared peptides. But if that a particular sequence of a protein, for example 8 or 9 amino acid length, a particular sequence of a protein is present to that only that protein, that peptide sequence you cannot find in any other human proteins, for instance, then that peptide is called as your unique peptide present for that particular protein. So when we are you are validating, or whenever we try in the global scales as well, whenever we are trying to get that peptides, or you try to identify particular protein, we try aim to look for the number of unique peptides. So that gives the confidence that the protein that we are looking for, or protein what we are claiming for, belongs is actually that particular protein. So the unique peptides of that particular protein, so we can monitor, or we can validate, or we can claim that our protein, whatever protein of interest is there, is only the unique peptide. So that's why whenever we are setting up an SRM assay, first we need to jot down or list down the peptides which are unique for that particular protein. So whatever proteins we are monitoring, or we are trying to monitor, we need to have the peptides for that. Then the peptide should be detectable by your LCMS analysis. So it should be detectable by your different LCMS analysis. Then if your peptide should be of 5 to 25 amino acids. So this is a criteria for your particular, whenever you have large peptide and large ions, it becomes difficult to get identified. So that should be of optimum length to get detected in the mass spectrometer. And the peptide, so whatever peptides, obviously you need to have understand that if you are using the whatever proteolytic enzymes you are using, that peptide should have, the protein should have those cleavage sites to generate that particular peptide. Avoid missed cleavage sites. So what is your missed cleavage? So the enzyme trypsin is not always 100% efficient, and what it does is that sometimes uh, it doesn't cleave at a particular site. So generally in a global proteomics data sets, whenever you try to do a primary data analysis, so what standards in the, according to the proteomics community standards, generally what is followed is that up to two missed cleavages are allowed, and two missed cleavages are used, so that you are allowing uh, your algorithm to detect the peptides where trypsin has missed up to two sites, and still it will identify for your particular peptide. But generally, in whenever you are setting up, so these are some of the differences between your global proteomics analysis and setting up your SRM MRM assay. That certain uh, cleavage sites you will avoid. So you will be using here zero missed cleavage sites. Then you will have two enzymatic sites, rank dents. You should avoid your rank dents, which says that the two enzymatic sites which are sequential to each other, and avoid frequently modified amino acids like your methionine oxidation, glycosylation, etc., etc. And also, whenever you are doing whenever you are trying to identify and analyze your particular proteins of interest, so what is the thing is that you should try to find out for the known mutant modifications like your signal peptide, isoforms, cleavage site, etc., to get an idea that how you are going to try to monitor this particular peptides and the proteins. So this is quite important steps. So first important thing is to be that it should be unique. It should be easily detectable. It should have a length. You should avoid missed cleavages. So missed cleavages are generally allowed in your global studies, but not here. And some drag dents and the sequential, so that you want the cleavage to happen, and you want to, for example, if you have a particular protein's interest, so you need to validate as many peptides of that particular protein possible, so that you can be highly confident about the result and the understanding. So now second is your peptides fragment selection. So as we mentioned that in the SRM or MRM, so now till now I think you are clear that we are selecting both our precursor ion and the fragment and their MYZs, precursor product MYZs. So how is our peptide fragment selections? So specific to our peptide of the interest, select Y ions with higher MYZ, because it's sometimes the solvent ions in the low sector in the low MYZ. So we select the Y ions. So I am hoping that you all are aware of the ABC and the XYZ ions, that where does the Y ions formed. So Y ions are formed by your cleavage at the C terminal site, whereas similarly the B ions are uh, cleaved at your N terminal site. So these have all been discussed in detail in different previous weeks. What are Y ions and B ions? So these are all the concepts and the basics through which we need to try to find out and try to understand that how we can do that. So now B ions are often of low abundance. So there are many reasons why your B ions are low abundance, because we are using C terminal proteolytic enzymes, and this remains quite frequently absent in your triple quadruple fragment spectra, and MSMS signal intensity. So it it is important for you to note that the intensities of individual fragments derived from one precursor ion differed substantially. To obtain high sensitivity assay, it is therefore essential to select transition specific for the most intense fragments. So whatever transition. So here, if you go, so that's why I had given the definition of the transition, so that you are able to understand and are on the same page, so that you can understand that what it is going on. So here it is saying that the fragments what you are trying to select for the transition, that is for the precursor and the product ions, that should be of high intensity, that to obtain a high sensitivity assay. So if you want to develop or obtain an assay which is quite sensitive and high sensitive in nature, so therefore it is essential to select transitions that is specific for the most intense fragments, not less intense but more intense fragments. So now another thing that you need to optimize for each transition is your collision energy. So here, so all these, so if you see in these experiments, so all these steps, firstly your sample preparation, followed by enzymatic digestion, creation of that particular peptide, then the selection of the pairs, and then and using these two quadruples and the collision energies to generate the fragments which will be of your interest, which will you would be able to detect, is quite necessary and quite important and quite you have to necessary. So that's why this step is again important, is to optimization for each transition to maximize signal in response or sensitivity, that is your collision energy. So what it is that optimizing your collision energy for each transition may increase your overall sensitivity, because if you are for each particular fragment product and your ion pair, if you are trying to optimize your collision energy, so it will increase your overall sensitivity. Okay, so however it is really needed, so it is not enough practical, and it is really needed when your sensitivity is very low or below the limit of detection. So there are two limits in a instrument. So one is your limit of the detection, that what limit, at what abundance it gets limited, and what limit it gets quantified. So one is your limit of detection and one is the limit of uh, quantification. So here you can see that the base collision energy on the TSQ voltage, which is given on the instrument from thermo, the thing is that how much is your M by Z's and how much is the collision energy is calculated. So this is how the your calculated for particular M by Z pairs between your product and fragment ion, so that you get the the fragmentation happens in such a way that you get the maximum fragment ions, intense fragment ions for the selected particular precursor, and that you can detect in your detector. And this is all the base collision energy based on your TSQ quantifier, based on Skyline, which we will look that how to use that. So, so how do we do the validation of the mRNA assay? So to confirm peptide identity, so you need to identify your peptide identity by acquiring a full MS2 spectrum of the peptide in that triple quadruple instrument used for mRNA. So you need to acquire a full MS2 spectrum of the peptide. So that will confirm your peptide identity. So I think we have also discussed that why we need to have a MS spectra to identify the peptide sequence, because only one MS spectra will give you the peptide fragment, peptide fingerprint, peptide mass fingerprint. But if you do the MS, then only you will 2MS will give you the peptide in the triple quadruple instrument. Then you will, how we will extract selected peptide and the charge state peptide fragments. So another important thing is to that the charge state, I think in the ESI, then the peptide fragments, corresponding M by Z ratios, fragment intensity ratios, collision energy for each transition, and chromatographic elution time to optionally use in scheduled MRM analysis. So that how it is getting eluted, what time the peptides on the basis of a gradient. So this is uh, quite important, and to understand that will depend that how much robust your analysis is and how you can try to find out. Now another thing is that, as I told, that you cannot try to find out and try to monitor each and every protein or each and every peptide that are present, that so you need to have a peptide and protein of interest which you are trying to find out from different uh, methods, like your uh, we're trying to find out from this short list from your using machine learning based methods or using your statistical methods to have a set of a particular proteins or a particular peptides. So but what you will do is that you are trying to now monitor, so how many transitions and can I monitor? So this is also an important question, right? So which I am sure that have come up in your mind, that how many peptides or proteins or how many transitions you can monitor. So we had discussed in detail regarding dual volume, dual time. I am hoping that you all are able to recall. That's why we have gone step by step in these sessions, and we have discussed, and in your course, which we have in this, we are discussing step by step with all the concepts, so that you are able to understand. So here you know that the dual time, if you are not sure or not clear or not able to find, then please I would say that go back to that particular videos and then try to find out the meaning of those. So this is your, for example, dual time of your 20 millisecond number of transitions. So if you have 10 peptides with the 10 fragments, will give you the scan time. So scan time of 2 seconds. So if you have a dual time of 20 milliseconds, number of transitions are 100, and you can have a for a 2 second scan time, it will have 15 points per tick. So then as the number of data points will be having in the peak, so it will give a much more sharper, so it will able to detect the if high confident, and you will have a good peak shape and size. So that's why it is important. So now that how many time you would give the detector your instrument to scan your particular ions or peptides. So here what is saying is that with a dual time of 20 millisecond, 100 to 150 transitions can be monitored with a 2 to 3 cycle second duty cycle. So if you are trying to monitor greater number of transitions, and if you do not give sufficient scan time, so now it will depend upon your instrument scan time available. So if you are not giving enough scan time, then what will have that the data points that you will receive, it will get reduced. So if your scan time your instrument doesn't get enough of the scan time or enough of the time that it can able to detect this, then your data points will get reduced, and what will happen is that your peak shape won't be so good, and you will not get so much confident results. So that's why it is important to understand that how many transitions I can monitor for a particular duration of the time. So 10 fragments per peptide, 10 to 15 peptides per LCM is done with 10 to 15 points across the chromatographic peak of about 13 seconds. So that you can monitor. So 10 to 15 peptides you can monitor in 30 seconds with very good quality data with 15 points per tick, if you have this number of transitions, and if also you need to understand your dual time of your instrument. So on this basis, now you can, if your runtime, if you are how much you can increase your runtime and how much you can go through it, on that basis you can try to find out, calculate that what how many transitions you can monitor in your particular run. So this was about the selected reaction monitoring or SRM assay. So we will be looking at the software Skyline, that how that transitions, what we are saying is that how we can prepare this transitions line transitions and how to set up in a format for a particular protein. So we will look into that. So now I think it's so any question for selected reaction monitoring or multiple reaction monitoring? Any question or any query till now? No, right? Okay. Okay, so we will move into the now PRM or the parallel reaction monitoring. So, so the steps. So first thing you need to remember that the instruments what we use is quite different. One uses a triple quadruple setup, another uses your uh, orbit trap setup along with the quadruple. Second thing is that the peptide length and the that things will remain common for both because you need to identify the peptides. They should be unique in nature, so it is always valuable if only you get uh, unique peptides you monitor, then only it is fruitful or useful, otherwise it is not so much use. Second thing is that second what is the thing is that you will be having all those miscleavages, the sample preparation, everything. So now here you are doing you are giving the separation and you are saying all the ions to get detected. So here you can see the fragments, these are the intense fragments, there's so many fragments are generated because with one fragments it is trying to identify all those fragments in the orbit trap. So this is the simple mechanism of PRM. So what is you can see that it performs a full scan of each transition by a precursor ion parallel monitoring. So why this name? So with the name only you can now I think you are now able to visualize and understand that what is the why this name single selected reaction monitoring or multiple reaction monitoring, or why this name of the parallel reaction monitoring. So it does the parallel monitoring of all fragments from your precursor ions. So whatever precursor ions you are going and giving, so it is what it is doing, it is parallelly monitoring all fragments from your precursor ions. So that's why it is the name was parallel reaction monitoring. So it uses your quadruple to select the precursor ion, and the precursor ion is fragmented in your collision cell, that is your Q2. So that in this collision cell, the precursor ion is fragmented. So in place of orbit trap, so sorry, it will be the place of Q3 by orbit trap. So where in the selected reaction monitoring you are getting the quadruple, here you will getting the orbit trap. And it what you will do is that you will scan all your product ions with high accuracy and resolution. So you will so Orbitrap generally provides you a better resolution, so it will scan all your product ions with a high accuracy and your resolution, which lead to your mass accuracy can reach at the ppm level. So again we had discussed, I'm hoping that you remember that the mass accuracy is your theoretical mass spectra minus your observed mass spectra into at the 10 to the power 6. So that is at the ppm level, and no need to optimize the fragmentation strategy. So there in the SRM one thing you are saying that you are need to having a collision energy because you are selecting a particular fragments. So what you are doing is that product and the mass pair. So if you are going for a particular product and the mass pair, that the precursor and the product pair, so you need to have your collision energy to generate that and then detect in the detector. So that's why it is there. But here you are analyzing all those fragments and all those fragments at a go. So that's why you were saying that it is no need to your optimize your fragmentation strategy. Okay. So now we'll look into the Skyline, that how we can generate it. Okay, so this is, you can find out, you can type in the Google as well, Skyline, Delhi. You can find out and install and do it if you are very much keen to do it. Uh, Otherwise, so what is there is that, so this is how the interface generally looks. Don't So I'll just uh, go to it. So now this is the interface of your Skyline. Okay, so what you will do is that, so if you go to edit and then you say that insert. So here you can see that the FASTA proteins, peptides, or the transition list if you have made it first. So let us look. So for example, you have protein. So for example, you have want to insert, you want to monitor albumin, human albumin. Okay, so if when I am searching, you can find out there it's coming albumin human. I will enter. So you will check for errors. So it will give your no errors, and then what you will do is that you will give insert. So now what you can see is that it is giving the peptides. This is your precursor. For, so here if you just go and see here, you can find out. So this precursor charge, that is a 2, precursor m by z, precursor m plus h. So in the positive mode m plus h, spectrum count, the ions, y ions. Okay, y1, 2, 3, 4, 5, 6, 7, which gives your, so see the concepts we had discussed in mascot and all, that how the ion peptide sequence you can find out. So you can see that the y1, y2 minus y1 and the b1. So from how the sequence is derived. Okay, for the peptide sequence, and it will give your transitions y1, y2, y3, y4, y5, 6, y7. This is your precursor product, your transitions. So, but before finding this, so you need to have some settings. So, if you go to the settings, then what you will do is that you will go to the peptide settings. Okay. So I am hopeful that you all are clear till now. So, in the peptide settings, what you will keep is that digestion by trypsin. So here you can see that these are the list of the proteolytic enzymes that you will get, that one can use. So what we generally, or what is very much popularly used, one is trypsin, another thing is lysine is also used, but mostly most labs uh, throughout uses trypsin as your digestion enzyme. So you can see here trypsin KR, not pro P. I am hoping that you all know. If you don't know, then what is the meaning of this? Is trypsin. It will cut after lysine and arginine in your sequence, in your protein sequence, except if proline is not present. So that not proline. So if proline is there immediately after lysine or arginine, then what will happen, that it will, trypsin won't be able to cleave. So the peptide list for a particular protein, sorry, whatever you are saying is cleaved. So the software, what it is doing, so the software is doing is that, so that's why so I'm giving you all a quite a hands-on kind of with the theory, so that you can understand. So the so as you know that we are now setting up, if you need to set up this experimental level for the SRM or MRMSS. So now what we are doing is that it's instructing the software that theoretically digest all my peptides on the proteins on the basis of wherever you find K or R. But if the P is present immediate after K and R, you won't digest that. Okay. So maximum miscleavages. So now only we discussed that the maximum miscleavages we should be 0. In the global data settings, in the primary data analysis, we can keep up to 2 miscleavages, but what we generally do follow So it gives you the option to have the miscleavages, but we are giving it as a 0. Then what is that? So here, so since you could see that I typed the protein name and it gave your particular protein, right? Albumin. So how did it know? So because I had already entered into the background human proteome as the human Swissprot proteome. So whatever we have downloaded from Uniprot human, so we have already added to it. So it is the same concept as in the mascot, as we have seen that we are trying to get the peptide sequences and matched. So the in the theoretical spectra along with the in this spectra, and now we are predicting it in an opposite direction. So for example, if you now, since you all have done mascot and know any other software or tool for primary data analysis, so now in the mascot what it's done, so you have your protein ions have been generated in the mass spectrometer. Now you are going to trace it back. So for that, what you are doing for tracing to back, you are generating a theoretical spectra. You are taking the fragment sequence and matching your experimental spectra and trying to produce the result. Now here what we are doing, here we are taking the proteins, leaving it in the peptides, and then we are make the product fragment pair, and then we are we will make out the our transition list, and then instruct our mass spectrometer to detect that whether check that these transitions that we are telling and give us the result that whether it is matching or not. So it is in the opposite direction. I am hopefully that I am able to make it clear that here what we are doing is we are predicting our, not predicting, rather we are making the precursor and the fragment ion pairs. Accordingly, we will give the instrument on the basis of the theory. We will give the instrument that we will select this particular precursor and find out me out these particular fragments. And what we will do is that the instrument will give us the result. Whereas in that primary data analysis, we have already generated in the global data, what we have done is that we have already generated the ions, and now we have the data, and then we trace it back that which proteins have been identified from the database. Now there is an important thing is that we are enforcing our peptide uniqueness by, so that's why I said that we need to have the uniqueness. So our peptide should be unique. So here the software gives you uh, a choice or the option so that you can enforce your peptide uniqueness. So here you can just go and here you can enforce your peptide uniqueness with protein. So as I was mentioning next part, so I will show you. So for example, you have got so I'll just come back. So I will show there also. So it's always a good idea to one second. So for example, so this is a peptide, right? DLG. So one second. First, I'll. So if I go to your export, so if we search proteins, sorry, here if you go here in the tools, you will find peptide uniqueness checker. Okay. So here you can give peptides. Okay. So we have already in the software enforced peptide by uniqueness, but you can always go and cross check. So what is the peptide is that DLGEE. If you write DLGEE, and NFK. Okay, so this is, for example, you can see, it is unique. How you identified here? You can see that if it is star mark, it is unique. If the star mark is not filled, it is pseudo unique. If this is not unique and identical sequences, so it is unique. So you can see that because we, in our software we have already said that we want only the peptides to be unique. So whatever peptides it has got are the unique peptides. So that right, you can cross check in the Nextprot tool in the peptide uniqueness checker as well. That's how you can, if you are able to get the unique peptides or not. Okay, so now let us go to the settings. So we are in the peptide settings. Okay, now is the prediction. So we are not predicting any retention time predictor. So these are all done if you have done the experiments, and for the, as I was mentioning, they are scheduled runs for those things. Yeah, so now this is important. So here what we are doing, minimum peptide length. So there, when we are setting up that experiment, there we had said that it should be of minimum peptide, some certain amount of length. So this is important. So this generally it is to be kept at 8 to 16, or you can tweak it accordingly, but it cannot be less than 5, not more than 25. So that you need to understand. Then N terminal amino acids exclude potential reactants. So that's why I have given all you the concepts to understand and use it accordingly, and exclude peptides containing, so you can make those filters accordingly. Then is the library. So what is the library? Is that the library is that the, as I told you, that there are two theoretical library and the spectral library. So if the fragments, so in the MS2 fragments, so in the, as I mentioned, that in the MS2 fragments, you are trying to find out that the peptides that you have matched on, how it is looking. So now, whether that you are confident or not, whether the peaks that you have got are relying or are reliable or not, that you can do with the library. So you can have a library. How you will can generate a library? So if you have run your same sample in the mass spec for the global mode, so you can use that mass fragments to make a spectral library, or you can do, what is that, you can, there is a tool called Proceed, which will, in using of the neural networks or the deep learning based methods, it will theoretically determine that what could be your experimental spectra can be, and then you can try to match with the MS spectra that you generate from your mass spectrometer, that how much it is matching with your library, to be confident of the peptide. So this is one. So you can say that library and filter, or library or filter. So any one you can try to find out. Then is your modifications. So carbamidinomethyl, we cannot do that anything, because that will come as in your sample preparation. It is coming in your uh, it will come when you do the sample preparation, uh, because of you are reducing it using TCP or DTT, and then alkylating it using IA for your trypsin to work on your proteins and cleave those. So that's why. And here I had said that there are LOQs and all those. So now we are skipping for this. So this is the simple standard modification that we can generally use. On the basis of that, what it has given is that it has given the list of the peptides for a particular. So it is unique. So I have already shown you how to go and cross check, and then you have the peptide list, and then the transitions. So as we mentioned that you will not, the B ions generally remain absent in your triple quadruple experiment, so you will get only the mostly the one Y ions. So now what you will try to do is that export what, your transition list, right? So here you will give the instrument name. So what is your instrument? So it can be of what? So for example, if we give thermoaltis, so which is a triple quadruple based mass spectrometer. So will you want to give single method, one method for protein, or multiple methods? So that you can decide. So one, if you give single method, so whatever transitions, so here you will look at that you are using two proteins, 44 peptides, 43 precursors, 418 transitions. So you can see that 43 precursors and product ends. So total transitions are becoming 418. Okay. So now you can use single method or one method per protein, or you can multiple methods. Okay, so you can order by M by Z or ignore proteins. So how many maximum transitions you want to give? Okay, so if you say 420, so now methods will be 1. If you say 200, then the methods will become 3. So you can keep accordingly. Okay, if optimizing none or collision energy. Okay, so that step also we had discussed, that how you can to optimize. So method type is standard, runtime duration if you for it run is your for 10 minutes. Okay. So now if it's coming, okay. So now I'll just transition list. So now with this transition list, what we will do is that we will ask the instrument to monitor these transitions, and if it is surely present in our samples, so what we will happen is that it will be get detected for transition list. So as if the peptide that you were saying it's present in your list, it will get detected, and once it gets detected, what we'll do is that it will get detected, and then you can do a relative quantification to find out that your trend with the global is matching or not. Okay, so this is a way you can try to find out and monitor. So now let us take one transition list. Okay, so now you can see that this is your compound, for example. So retention time minutes. So this is your precursor. This is your product. So for this precursor, you have this particular product, and this is a collision energy. So this you will fit into the instrument to find out for those particular proteins. So this is how a particular transition list generally looks with the particular peptide. Okay. So now I will show you one example that if you have, yeah, so as I will show you with one experimental data that how it looks generally. So after the experiment, how it looks, so that you get a feel of it. So for example, this is an experimental data. So these are your different samples which has come. So as I was mentioning the peaks, so how it looks. So it will, if you're have the peptide, so you have given the transitions. So if you look at it, so this is the precursor and these are the many transitions, products, precursor and the products that have detected. So if you give your some scan time sufficient time and it gets detected that particular on that particular collision energy the different peptides and the peaks, so it will give you. So this is that particular transitions, that particular ion point. So it depends, this peak shape depends on the number of data points that have been acquired in that scan time. So it will depend how much scan time you provide to the instrument. So that's why it is important to monitor that how many transitions we will give in one method in one list. So for example, here we are monitored 128 transitions. So this is a real experimental data. So you can see that all the transitions, you can see that with a good peak shape it is showing. And what library we were saying that, as I was mentioning, that this is the library. So it is being generated and it how it matching. So you can see that it is matching with the library that has been generated. The experimental spectra it is getting uh, matched with it. So this is how generally a Skyline file looks, and this is how the transition is uh, are made. So any question for this? Sorry, but I have repeatedly said that I cannot answer any these questions directly. But if you have any questions now, so this is how the Skyline files are made, and this what we are uh, what how we will get those make those transition list which we will feed, and this is how the final result comes, from where you can find out the results and then can do a relative, then again you can perform the statistical test which will give you the results that how much it is significantly dysregulated. So you can then check out the trend between your global data and the Skyline data, targeted data. So Skyline is a software that is being used, popularly used for this kind of data and to find out and make out the transition list. So hopefully there's no question. So now if we go for the public data storages. So now, if I go to the first phase of our presentation, so we have said that these are the two things that we will do. So we have looked at the validation of the particular proteins. Okay, so now both antibody-based method, but we have generally what we are doing is the MS-based method, how we can validate of the proteins or the proteins of the interest. Now what we are trying to look at the data storage and the accessibility. So now there the huge amount of data is generated. So now where to store and how it is stored, and from there you can access the data. So for different proteomics and the genomics data sets, so we will look into such repositories. One such repository is Fire Cloud. So you can go and try to find out in the Fire Cloud. So uh, why these so these things are needed. So before that also, these analysis takes, when you have a huge amount of samples and all you have to analyze, if you have a automated script and automated way of analysis, then it becomes quite easy for you to for change a certain step of analysis, or you need to identify certain sets of proteins and the peptides and those. So then what you are going to find out is that you can find out the different uh, proteins, but sometimes if you are so sometimes these many things have helped you in creating those scripts and the automated analysis that will help you to go into the fire cloud. Okay, not fire cloud means automated scripts and those for the analysis, from where you can easily try to find out those particular follow the analysis steps and get the analyze those results for different data sets. So one example is your fire cloud, which have been kind of developed on the basis of the, I think, Broad Institute of the NIH, where you can get the different project, thousand genomes project, where you can create those account and try to access those for the different analysis. Now there is one so for the proteomes and the data sets. I'll just show you all. So Proteome Exchange is one of those consortium which have all the raw data repositories. So whenever you do a experiment and sorry, so whenever you do an experiment and if you want to share it with in and publish in the manuscript, so you need to upload your raw data for the reviewers and the publicly access. So this is your Proteome Exchange. So this is the Proteome Exchange consortium was established to provide your globally coordinated standard data submission and dissemination pipelines. Main proteomics repositories encourage open data policies in the field. Okay, so this is the consortium where you have your peptide atlas. For your peptide atlas contains all the information of your, as I mentioned, of the transitions and the peptides that have come in the serum methods. Massive, right? Is your European EBI based repositories. Then, so as NCBI, I think you all know the NCBI and EB that EMBL thing, and the for the nucleotides, the nucleic acid sequences of all those organisms that is here. Similarly, is the Proteome Exchange Consortium, which contains all these iProgs and the JPost and the public data which you can access. Okay, so you can access all those Proteome Exchange data are generally given a Proteome Exchange ID, PhD ID, and then you can find out. So you can submit your data and you can access your data. Those these are the different platforms which are present. So if you go to the Pride Proteomic identification the database, so from here you can identify, you can download and analyze your uh, you can download raw dataset from Pride. Okay, so for example, if you go to this PhD ID, so if you have the PhD ID, so if you go through any manuscript, in any manuscript who have used the raw proteomics datasets, they have uploaded, and then you will find out the uh, uh, Pride ID, this PhD IDs. So you can try to find out this PhD IDs in the proteomics and then go to it. So here you can see. So here you can see this is the raw files. So if you just download these raw files, so you can get all those raw files, and from the paper and the metadata you can find out that which sample belongs to what. So whatever your interest, so whatever they have said in the paper, you can first try it out in your own method to find out that how reproducible it is, whether you are getting the same result, your pipeline is following the same result or not with the published data, that whether your pipeline is working fine. Second is that different types of analysis and analysis methods you can try to find out there. So this is how generally it works, right? So this is one of the major consortium, and in the Pride you can find out any proteome data, protein raw mass spec data, which you can analyze and then can find out the proteins of interest. And then in the Skyline you can prepare your transition list, and then you can run in the mass spectrometer to validate your particular proteins. Now, second, another thing is the omics DI. So now if you go to the omics DI, so here you can access. Okay, so here it contains many things. Okay, so for example, metabolites for metabolomics workbench, Metabolome Express, GNPS, then Pride, Peptide Atlas Massive, which I shown. Then you have the genomics, transcriptomics. We go for browse. So here you can see transcriptomics, multiomics, proteomics, genomics, metabolomics. So any kind of omics data set you can try to find out. Single cell transcriptomics, metagenomics, genomics. So if you go for proteomics, so here you can see omics type proteomics, autoimmune in BXD to mice. So all these things you can. So these are not only mass spec data but array data, proteomic profiling by array, microarray data. So you can find out all those different methods and different studies there. So if you go for multiomics, so you can do this exercise, go to these different websites, different also in the manuscript. If you come through, if you ever gone through some proteogenomics manuscript or the proteomics manuscript and have identified certain, come across to PhDs, go through that, go search in the proteomics exchange, search in the Pride, search in the omics DI, try to find out what are the data sets, and in case any type of these things. Another is your so here you can see your different things. Another is your array express. So yeah, so this I was going to mention this only. So array express contains your different data for your functional genomics data. All those in that you will have peptide microarray data, protein microarray data. You can find a lot of data in the array express as well, and it has also a bioconductor package to access array express data and then build up your data, and then you can perform those data. So all these things can be easily accessed. Okay, so now this is one thing. So now I'll show you another thing is the gene expression omnibus or geo data sets. So data storage and accessibility, and then so you can see, you can understand that there are so much huge quantity of data that is getting generated that needs to be stored safely, secured, and then you need to access those particular uh, data sets. So that's why it becomes quite crucial and understand. So here I'll show you one uh, kind of thing that what you can do is that, okay, so if you take one example, take is an expression profiling by array of the, if it's generally if you take your kind of a microarray kind of data, so this starts with a GAC number. So if you now I think if you come across through any manuscript and find out that if you find out PXD ID, then you would be able to search in the proteome exchange. If it is containing GAC ID, then you will be finding it in the geo datasets. Similarly, in the array express. So and also in the data availability statement of any manuscript, you will find out your data, and then accordingly you can go to that particular metabolites or anything and try to find out. So now this is a, for example, a geo data set. So now I will show you a simple easy way that you can analyze this kind of array express data. So for any disease, so whatever you are interested in, if, for example, this is for the B cell wild type mutant, disease control, tumor control, different grades, whatever you find out, you can easily for your expression data and expression analysis, if it is there, you can come and try to analyze. So here you will see that there is an analyze with the GO2R. So if you are not comfortable or do not want to go for the script, art script and all automated script, so there are tools. So what I try to intend here is that there are different tools and those available that help you will in analyze in a very automated, in a simpler way, to so that you can so that the curious scientists and the biologists or the researchers can try to find out the different available things. So here you can just click on the analyze with the GA2R. So what is that? So you can find out. So it is based on the hard script. So here what you can find out the accession number, the title, what is the wild type, samples, source name, EBV strain. So whatever type of analysis you want to do, you can do that. And the cell type, everything is given. Then what is saying that you need to analyze. Okay, so here R script is also available. So if you want to try to find out and do it on your own, that is also possible. So here you will go and analyze. Okay, so what you can do is that go to the options. Now apply adjusted to the p values. So I think we had discussed, might be in briefly, but that what is adjusted p value, we need to control our false discovery rate. So here you can use Benjamin Hollenberg. So generally in the proteomics or the transcriptomics or the biological data sets, we use Benjamin Hollenberg because the number of features are huge, and bond friendly correction would lead to a more stringent and increase in the false negative error. Okay, so this is one thing. Then if you want to apply your log transformation, apply lemma precision, force normalization, so then you can try to do a reanalysis. So these are all the parameters that you can tweak and try to find out and then perform the uh, reanalysis. So you can see so how your data looks fairly normalized, expression density, mean variance trend, UMAP plot. Okay, so you can using GO2R, you can easily uh, do your analysis. Okay, so this is another platform through which for your interest disease, interest organism, you can go and easily play and try to find out the data if you are not willing to do it uh, in a manner in a writing the scripts in any language like Python or which will help you in a automated uh, processing and all, so that you can easily do and check. Okay, so this is the different data storages. Now there is a tool or Galaxy server which I'll show. So this gives you a kind of a all kind of analysis pipelines which you can use it accordingly without any hustle and can easily perform. So for example, yeah, so this is the tool. So here also you can perform genomics, proteomics, metabolomics. So I think I had given certain things in the genomics, certain that you can import your FASTA and FASTA file. So here you can see that combine FASTA, concatenate, split FASTA, FASTA merge. Then you find out the quality check of those. So in the proteogenomics pipeline, this plays a huge role where you can find all your genomics data, where you can analyze your genomics data sets followed by proteomics data sets by making the pipeline and can try to analyze, but only that you have to understand that it works since it runs in a server, so it will have some issue with the time. So this is running all these tools which you can easily install and perform and analyze, but if you are not comfortable you can also use the different server. So because all these tools also require certain computational skills, along with that requires your because the data sizes are huge, it requires quite a bit of processors and setups that would support this kind of data. So these kind of tools like your Geo2R, Galaxy tools are quite handy in this manner that you can analyze in easily. Then the genomics analysis of assembly, annotation, mapping, variant calling, chip-seq, RNA-seq, all can be performed. Okay, so this all can be performed on a Galaxy. Okay, so high set 280 sec count. So all these things, there are tons of tools which you can do. Similarly for the proteomics, okay, custom prod, we generate protein faster from exosome or transcriptome data. So if you, so the proteogenomics, so if you have a BAM file, if you have a PCF file, you have a genome annotation, so it's G38. This is, if you have for human, you can create a particular database through which you can search in the proteomics data. That even the global data you are searching through the UniProt human, but you have a mutant database, mutant genomic sequence, you can easily get from here. Create a decoy database. So what is a decoy database? So once we are giving identifying the peptides and all the algorithms depend on the protein identification, but when we find that, if we find that some certain decoy database will create certain peptides having those spectra which are quite similar to the human proteins but are not of the human proteins, so that your false identification of the primary analysis becomes less. Then the DIA based methods. So all these you can find out here. So here you can use MaxQuant as well. So you can, if you want to convert any files, you can do that. Okay, image tracker. So you can find out all those things here. So this gives you a very good, if you, so in the Uniprot, what we do is that we download that the FASTA file that we generally employ or generally deploy in what that I was showing you in Skyline or in the primary database. So you get that particular files Uniprot. So here easily you can download your data and you can XML or the FASTA format, and you can have the Uniprot tool from here. It's in the Galaxy. So it gives you uh, quite a range of things. So you can try to find out also in the metabolomics, batch correction, transformer, camera annotation, and all these things you can try to find out in the in this, gives you are very much in a highly automated manner without having your much processors or different things. You can try to use different tools and try to analyze your data. So the Galaxy is very much useful in that manner, which help you to uh, identify those sequences. Yeah, so this is another tool. Yeah, so the mostly, so we have covered this week is that after we have got the global set of proteins, how we have validated and what are the different data storages. So most important is one of those your omics DI, one is your geo database, one is your proteome exchange, metabolites. uh, these are all very much important for your different omics, and omics DI contains your multiple omics datasets. And then you can also analyze using, so you have downloaded, you can use download your datasets from their online repositories without having you need doing any experimental setup, and then you can search in the different tools or servers like your Galaxy or your different algo your in the Geo2R. and can try to find out a preliminary data set analysis on the just on the basis of your curiosity, and then obviously you can trace it back with the literature to find out a biology behind it, and then can always try to find out the actual experiments and in the targeted method or other method that what is happening. Okay, so any question for today's session